Okay, so today we will be talking about DNA and protein synthesis. Hold on, I'll make sure y'all can see it. So DNA is a nucleic acid that was one of the four biomolecules that we talked about the first week of school. And DNA, I told you had two functions. The first function was to tell how we are genetically made up, so how tall or short we are, what our hair color would look like, what our hair color would look like, so on and so forth, as well as it holds all the instructions to create the proteins that are found within our body. So first, today we are going to talk about what DNA is, what DNA looks like, what the building block is, how it's built, and some relationships that you see in DNA. And then starting not tomorrow, but Thursday, we are going to talk about how DNA creates proteins. Okay? So, this will not go in your notes, but it's just an important fun fact. Did you know that if you unraveled all your chromosomes from all of your cells in your body and you laid the DNA out from end to end, the strands would reach from the earth to the moon about 6,000 times? Okay, so obviously, you know, from the Earth to the moon one time, that's an extremely far distance. But all of the cells and all of the DNA that is found in each of those individual cells, because we have trillions of them, if you line them up, it can go 6,000 times. Okay? And before I keep going, when I post a PowerPoint onto Google Classroom, look in the speaker notes because I've looked at some of your notebooks and a lot of y'all have notes that don't need to be there. Look in the speaker notes. I've put important or not important. If it says important, put that into your notebook. Okay, so like this slide doesn't have anything on it in the speaker notes. So that means you don't need to put anything from this slide on your notes. Okay, but it is just something that you need to know. So James Watson and Francis Crick, they were the ones who were responsible for discovering the DNA structure. They ended up winning a Nobel Peace Prize in the early 1950s for discovering the structure. However, they would have not been able to complete this work without a woman named Rosalind Franklin. They used her work without her knowledge or her permission, however. So, you know, women, we run the world, but men just take over. Yeah, it was happening back then too. Okay, so Miss Franklin, Mr. Watson, and Mr. Crick, they were able to discover that DNA looked like two threads twisted around each other. So it looks like a twisted ladder. And this shape is called a double helix. So on the left is James Watson, on the right is Francis Crick. In the middle, that is Miss Rosalind Franklin. And the reason why she's in the middle of this picture is because we would have not known what DNA looks like without her contributions, even though we don't see her or we don't hear about her at all. And it's kind of hard to see, but back here are different pictures of the DNA structure. I tried to turn the light off so you guys could see it a little better, but it's still hard to see. Okay, so now we're going to talk about how DNA is built, its monomer, how the monomers are set up, and the bonds that are formed in DNA. Okay, so like I told you guys on the very first week of school, DNA stands for deoxyribonucleic acid. So how you pronounce that word, just break them apart. D, then oxy, ribonucleic. It's hard when you say it for the first time. But like I said, by the time you leave my classroom, you'll be able to say this like you can say, my name is so and so. Okay, so DNA controls cellular activity by influencing the production of proteins. DNA is a very long chain polymer. So poly means more than two, excuse me. So it's a polymer made up of thousands of repeating units called nucleotides. And you guys should remember that nucleotides are the monomers or the building blocks for all nucleic acids, both DNA and RNA. DNA nucleotides are made up of three parts. It has a phosphate group, it has a sugar, and it has a nitrogenous base. And nitrogenous just means that it's made up of nitrogen. Okay, so on the top, you have the phosphate. You can either see it represented by a circle or not a star, but okay. I forgot what that shape is called, but there. Okay. So it's either represented by a circle or a sideways star. It has a sugar. The sugar that is found in DNA is called deoxyribose. Remember, I told you all sugars end in OSE. So glucose, lactose, fructose, whatever. So the sugar for deoxyribonucleic acid is deoxyribose. Okay, and then this rectangle that is on the right, that is the nitrogen base. So like I said, the name of the sugar is deoxyribose. There are 4 different nitrogenous bases that you may see. So I'm gonna ask you guys to take yourself off mute because I'm gonna say the base names and then I need you to repeat them back to me, okay? The first base is adenine. Adenine, thank you, sir. Adenine. I heard it. I don't know who said it, but somebody said it. Okay, so the first one is adenine. Adenine, well, before I finish going on, but all of these bases, you will either hear me say the real name or you'll hear me say the letter. Okay, so adenine is represented by the letter A. The second base is thymine.
Thymine.
Thymine, like me. Thank you, sir. Like your thigh is me. That's what it is, thymine. Okay. The third is guanine. That was tremendous. Thank y'all for participating. I'm gonna give y'all extra credit. And C is cytosine. Thank y'all. So again, you will either hear me say the full name, so adenine, thymine, guanine, or cytosine, or you will hear me say the letters A, T, C, and G. I need you to know both of them though, both the name and the letter. So DNA nucleotides, they fit together with the phosphate and the sugar, creating the backbone, or what I would call the sides of the ladder. And then the bases, they pair together in the middle of the structure, creating the steps or the rungs of the ladder. Okay, so imagine this is one side of the ladder. If you were to flip this and put it on the other side, that would be the other side of the ladder. So in the middle, you would see 2 nitrogen bases, you would see another base here, and then on the side here, you would see another sugar and another phosphate. Oh, I had a picture of it. Why did I do that? So this is the polynucleotide chain because it's more than 2 nucleotides. It's 1, 2, 3, 4. Okay, so the sugar and the phosphate and sugar, those make up the backbones or the sides of the ladder. And then the nitrogenous bases, are in the middle of the ladder. Those create the step. So this part is important. The base adenine, so A, always pairs with thymine, T. The base guanine always pairs with cytosine. There's 2 ways that you can remember that. Okay, so the first way, my first group of students made it up. So they said ATGC, at Golden Corral. Okay, So I don't know why they like golden corral, but that was just how they remembered it. The second way to remember it, if you write the letters A and you write the letter T, you have to make a cross. So I'm a visual learner, so I gotta show y'all visually. Sorry. So when I write the letter, what am I doing? Okay, there we go. When I write the letter A, I have to cross this line. I have to cross over it. When I write a T, I have to do the same thing. G and C, I don't have to make a cross. They're curved letters. I don't have to cross anything. So you can remember that the letters that you have to draw a line over, they go together, or draw a line with, they go together. And the ones that have curves, they also go together. So A and T go together, G and C goes together. If you come up with another way to remember it, that's perfectly okay. Feel free to share. It might be easier for your classmates to learn. But as long as you know that A and T go together, G and C, oh, A and T, that's a school. GC, Guilford College, that's a school. See, as long as you remember A and T and G and C go together, that's all that matters. So these base pairs, they create the two halves of the double helix, and the strands are complementary. So what that means is, I have on a black shirt and dark blue jeans. Let's say somebody came in here with black pants on and a dark jean shirt. They would be my complement. We have on the same thing, but it's just an opposite fashion. So if I saw a DNA strand where I saw ATGC, its complement would be the opposite. It has the same letters, but just in a different fashion. So TA, I said GC first, so then CG. If I say CG first, then GC. As long as it's the opposite. Does that make sense? Everybody else, does that make sense? Yes, no, maybe so? Cool. All right, so now we're gonna go over it together. So, what is the complementary DNA sequence for CATTGC? I'm gonna give you a minute, think about it. Remember, the opposite goes with it. So A and T goes together, C and G goes together. Give you a minute to think about it, and I'm gonna call on somebody to give you the answer. Jamaria, what's the complementary DNA sequence for this, please? You're in love. Somebody want to help her out. I think she's not here. Nobody? Would y'all like to do this one together?
Yes.
Okay. Thank you. All right. So now we're going to try. Yes. Okay. The DNA sequence given was CATTGC. So I told you A always pairs with T, G always pairs with C. So when you're trying to find the complement, you're trying to find the opposite side. So remember, like I said, I'm having black pants and dark blue. I'm looking for the dark blue and black. So if C was the first letter, what pairs with C? G. So G would be the first letter here. Oh, I don't want a black marker. What pairs with A?
T.
What pairs with T?
A.
So because it's a second T, we have a second A. What pairs with G?
C.
C. And what pairs with C?
G.
And y'all just made your first complementary DNA sequence. Okay? Does that make sense now?
Yes.
We're gonna try the second one by yourself then. So the second DNA sequence says GTACAG. So I'm still gonna give you a minute to think about it. This time I'm not gonna call you, I'm gonna ask for a volunteer. Maybe I wasn't ready. Somebody got it for me? Okay, what is it, Jada?
C.
Mm-hmm.
A. T.
Mm-hmm.
G.
Mm-hmm.
C.
Yes, ma'am. Perfect. I got one more for you. Or for whoever. GAGTAC. What is the complement? Somebody got it for me?
CTC.
Yes. Keep going.
A.
Mm-hmm.
GT.
Yes, sir. Perfect. Y'all just made the complementary DNA sequences. Okay, maybe not. Oh, there you go. So all that pops up are the answers that y'all already gave me. Right. And so these bases, they are held together with what is called hydrogen bonds. So in this picture, when you look at it in your notes, if you can't see it, you'll notice that there are different numbers of bonds between each base pair. So between C and G, there will always be 3 hydrogen bonds. There it is. 3 hydrogen bonds. And in between thymine and adenine, there are 2 hydrogen bonds. Okay, 2 red lines, 3 red lines. And lastly, this ladder structure is twisted to form a double helix molecule, and it is the shape of the molecule that allows DNA to tell an organism cells what to do, when to do it, and how to do it. Okay. So this is the last slide for today. I'm going to upload the notes. I'm going to upload the recording of the notes, and then I'm going to upload an Edpuzzle assignment about what we just talked about. And that will be your homework for tonight. Okay. So before I do that, before I log off for y'all, let me make sure I have everybody for attendance.